session, Dr. Beth Reinke, who studies coloration from a very integrative perspective, looking at both the function of coloration and also how selection pressures can shape coloration across species. So she got her bachelor's degree from Indiana University and then went on to a PhD at Dartmouth with my academic older brother, Ryan Kalsbeek, studying the function of coloration of freshwater turtles. She's now a postdoc at Texas A&M, studying how selection pressure shape coloration on both the dorsal and ventral aspects of butterfly wings. So please join me in welcoming her, talking about drivers of color diversity, interspecific and intraspecific variation in butterflies and turtles.
Thank you. Sorry, I'm trying to get the pointer to work. There we go. So I'm going to be talking about interspecific and intraspecific variation in butterflies and turtles. And this is two distinct projects that are tied together in both cases I was investigating the drivers of this diversity. But I'm only talking about interspecific variation in butterflies and intraspecific in turtles. So if you came for the opposite, you can leave. So interspecific color diversity, especially when we're talking about colors that is a visual signal, is often driven because it has to provide receivers with species-specific information. So we have these really nice illustrations by Ernst Haeckel of hummingbird signal diversity and orchids. And the system that I wanted to investigate interspecific diversity in is butterflies, and they make a really great model, not only because they're obviously very colorful and most of their body surface is a signal, but also because they make this nice two-dimensional color signal where we can kind of try to tease apart the effects of natural and sexual selection. So generally speaking, the dorsal side of a butterfly is used to signal to conspecifics or to other butterflies to defend territory or to attract mates. And the ventral side, which is what's visible when they're at rest, it has anti-predatory functions or maybe cryptic. And again, this is really generally speaking. So the group that I chose to investigate interspecific diversity in is Nymphalidae, which has this really nice phylogeny constructed by Nicholas Wahlberg's lab in 2006. And it contains something around 50 species, which are really diverse. So these are only 20 of the 50 species. You can see a huge variation in the types of colors they have, the patterns they have, and the wing shapes. And this holds true also for the ventral side. So both the dorsal and the ventral side has a wide amount of color variation. And when thinking about how to best investigate on a macroevolutionary scale how this color might be driven, I chose to investigate colorfulness. And colorfulness itself, I broke down into 3 different traits. However, as we all know, color is really dependent, especially when it's a signal, is dependent on a receiver. So I collected reflectance spectra of every little color patch you see here on both the dorsal and the ventral side, and then modeled those within receiver spaces. So as Natasha taught us earlier, birds are highly conserved, and they tend to, you can pretty much split them into 2 vision types, ultraviolet sensitive and violet sensitive. So I used averaged spectral sensitivity data for both of those. And conveniently for me, this group of butterflies, the Nymphalidae, are one of the few butterflies that are trichromatic, as far as we know. They're not one of the complicated ones with 8 different photoreceptors. So in that way, I was able to model all the reflectance spectra I collected from the dorsal and ventral sides of about 50 species in each of these color spaces and use those data to calculate 3 measures of colorfulness. Now the first, and to me the most intuitive, is color volume. So this is the minimum convex polygon that encompasses all the points within a color space of a given species. And again, I broke it up by dorsal and ventral and looked at 3 different receivers, the 2 birds and a butterfly. I'm probably going to say color volume a lot of time, but when this is modeled within the butterfly space, it's actually an area. The second measure of colorfulness is maximum hue disparity, and this to me is the least intuitive. But this is on a scale from 0 to 1, where 1 is that they have a large amount of hue disparity and the colors are directly opposite each other on a color wheel. It was just how we think of it. Whereas the closer to 0 you get, the more similar the colors would be if you were to put them on that color wheel. So I measured the maximum hue disparity for every species within each color space also. And then the third is the average span. So the span is measured as the Euclidean distance between 2 points. So I measured all the distances between all the points within each color space and constructed the average and used that as a measure of colorfulness within each of these receiver spaces. So here's an idea of what that might look like within each of the spaces. These are just 2 species, Aglae certicae and Anarcha emathea. And within the 2 bird spaces and the Nymphalidae butterfly. So this is like if you grabbed the U with your finger and pulled it toward you. The M's here at the bottom. This is the medium wavelength photoreceptor. And the dorsal spectra are modeled in black, and the ventral are in gray. So you can just kind of get an idea of what these data look like in a couple of the species and how the dorsal and ventral might separate in some, and how these little patches are formed in others. So then I was able to take these data and the colorfulness variables I extracted from them and plot them onto a phylogenetic tree, thanks to that nice tree by Nicholas Wahlberg's lab. So here's an example of what one of those looked like. This is color volume as viewed by an ultraviolet sensitive avian predator. And on the left, this tree is the dorsal side, and on the right, it's the ventral side. And then the color scale goes from low to high color volume. So right away, some species might stand out. You see this guy here that has a really low color volume on the dorsal side and a really high one on the ventral side. And there's an opposite one up there. But in general, by using this in ancestral state reconstruction, I can then fit different models of evolution onto this tree containing colorfulness and see what data I can get from there. So a quick review of some of the different models of evolution. Brownian, which is also known as the random walk model, says that the amount of variance in a phenotype distribution increases proportionally with time. So we've got time on the x-axis, some random phenotype on the y. The mean is going to stay the same through time, but the variance increases. Another type of Brownian motion is what's called Ornstein-Uhlenbeck stasis model of evolution. And this is, again, Brownian evolution, but with what's called a rubber banding effect, where there's something that's pulling the variance inwards toward an optimum. So that rubber band is going to be measured as a parameter in the model called alpha, which I'll talk about later. And what this might make you think of right away is stabilizing selection, but I want to point out that it is not equivalent. It's in some ways equivalent, but it is not equal to stabilizing selection because we're talking about a distribution of species that are all related. And this might be constrained not just by selection, but by intrinsic factors as well, like we just learned from Julian, that prevent that type of colorfulness production or whatever. Then we have Ornstein-Uhlenbeck directional. So again, this is the Brownian motion with a rubber banding effect that's pulling that optimum to something different than what it started with at time 0. So in this case, it increases. It could also decrease, go in any direction. And finally, the early burst, which is a little more self-explanatory. This just says that there was some wide radiation early on in time, and then that variance was maintained through time. So forgive the table, but it's actually pretty simple. So this tells us we're looking at here the 3 different colorfulness traits within each visual system, the dorsal and the ventral side, and the model that had the lowest AICC score. So in most cases, Brownian motion was the best type of evolution, or the best model of evolution to fit these data. In some cases, and they're in blue here, Ornstein-Uhlenbeck stasis was actually better. So here we're looking at hue disparity of the dorsal side as viewed by a violet-sensitive avian, and then 3 different traits, hue disparity on the ventral, average span on the dorsal and ventral, as viewed by a conspecific butterfly. And I guess I didn't say this earlier, but I only had data for one of these species of butterflies. They are trichromatic, and we don't know much about their vision, so it's hard to say how generalizable this is, but it's enough that this came out to be not just a Brownian fit. So this is in contrast to another study that did a similar thing, started in Prum in 2008, looked at colorfulness in buntings, and they found that OU directional was the best fit, and hypothesized that it was primarily driven by female choice. So we found in most cases Brownian is the best fit, with some OU stasis being a better fit. We can also look at the evolutionary rates, which are denoted by sigma squared here, and the alpha values. So again, I'm sorry for the giant tables, but I just want to point out a few things. In many cases, especially for volume and hue disparity, the rate of evolution is faster on the ventral side than the dorsal. And then also, if you pull out these alpha values, the higher the alpha, the stronger that rubber band, so the more the variance is constrained to the mean. And the highest one we see is on the ventral side, as viewed by the butterfly, the hue disparity. This is a little bit surprising when you think about what we know about these generalizable conclusions about dorsal versus ventral and who the receivers are. Where here we're seeing that it appears there are some constraints, some pretty strong constraints, as viewed by a butterfly on the ventral side. This part makes a little more sense. We see that the evolutionary rate is faster on the ventral, as viewed by a bird, which might be what we expect if predation is what's driving the colorfulness there. So it's hard to say what the actual concrete conclusions are of this study, but it does help us get at some of the drivers and some of the, by looking at the models of evolution that fit this colorfulness, it can tell us a little bit more information about the receivers. So part of the reason that I think it's hard to really pull out natural and sexual selection, as I'd originally hoped, is that there is such wide diversity here. So again, this is the dorsal side, which I had said earlier is primarily used usually to signal to conspecifics, and yet we have some with eye spots. We have a wide variety of colors, and then if you look at the ventral side, we've got iridescence here, bright colors here, iridescence there, a wide variety of colors that make this harder. So I'm currently in the process of kind of looking at this on a more species-specific level and incorporating discrete traits as well, like aposematism, cryptic, the presence of leaf-like ventral sides, and territoriality, all of which might also play into colorfulness. So to change gears a little bit and talk about intraspecific variation, intraspecific variation in color might be expected to be driven by environmental or geographic differences that impact the efficacy of signal transmission. So these are some cichlids where A and B are the same species and C and D are the same species, but these guys are in a different environment than B and D. And the lake water of A and C is more turbid, and so the colors vary. The color varies in order to better transfer the signal through that environment. Another explanation for intraspecific variation in color could be a just complete relaxation of selective pressures that leads to a predictable amount of variation, like if that color is not necessarily a signal. So here we've got some salamander head patterns where we just see these different morphs that are more or less discrete, but seem to have, as far as we know, no signaling function and no adaptive function at all. It's just due to a relaxation of selectant pressures. A third case, and this is one that Molly mentioned this morning, is this really extreme version where these different populations all have the same type of signal. They're all aposematic, but they're divergent and driven in different ways by female choice, most likely. And this is, I would say, a pretty drastic and rare, extreme case. So the case study I want to talk about today is the painted turtle, which is a species that most of you are probably familiar with because they're really common in North America and especially in Michigan. And they have this really bright ventral side, and this is called the plastron. However, there's a lot of intraspecific variation. So here in Michigan, we've got these guys, which have this bright red on the marginals, but not so much on the actual plastron. But if you go west to Wisconsin and further west and up north, they're bright orange. Moving east here on the East Coast, we've got another with some red. It's not nearly as bright or not nearly as high contrast, I guess, in general, as these two versions, but it's still there. And then the southern guy down here, which is pretty much just yellow and black, no bright orange coloration. So I was really interested in what could explain this kind of extreme type of intraspecific variation. I also want to mention that in the hybrid zones here, it's a continuous variation. So these aren't discrete morphs at all. There is quite a continuum. So in order to think about what could explain this variation, the first thing I wanted to know was, is this a signal? And if so, how is it produced? And so in thinking about how organisms make color, we know that they can make it through structure or pigment. And if we're talking about pigment, there are a lot of steps in this process. As we just heard about these complicated pathways in flowers. This is a obviously really simplified version where this box might represent an organism's body. So a pigment can be synthesized within the body, metabolized, transported to the tissue where it's going to be expressed. In another version, it has to be acquired from external sources and then absorbed through the intestine, transported to be metabolized, and transported to be expressed. So one of the first steps I took with painted turtles was to identify, using HPLC and absorbance spectrometry, what the color mechanism is. And I found it was carotenoids, which is pretty much the most exciting thing I could have found, because they cannot be made by vertebrates. So carotenoids have to be obtained from the diet. And in this case, carotenoids aren't typically limited in freshwater ecosystems. They're made by algae. They're really common in plants, and so a lot of macroinvertebrates obtain them from their diet. And an omnivore, like a painted turtle, should be able to pretty readily obtain carotenoids, but then they still have to absorb them, transport them, metabolize them, and move them to this keratinized matrix in the shell, which is where it's stored. So this suggested to me that this probably is adaptive and probably has some sort of signal function, because why else would they go to all this trouble of trying to get these carotenoids, taking them through all these processes, and then expressing them in their shell? So one of the first, let me first tell you about one of the reasons that I decided it probably wasn't a sexual signal is because there's no sexual dichromatism. So there's no difference in males and females in this orange color. And while that doesn't mean it's not a sexual signal, because sexual signals can evolve in both sexes, there's also no associated display of this ventral side. So when they're courting, the males and females will face each other underwater, and males actually have these really long X-Men-like fingers, and they wave their nails in the female's face, and that's how she decides whether she's receptive. But in that process of facing each other in the water and waving, there's no display of that ventral side at all. So if there's any viewing of the orange color, it's pretty incidental, and it's not associated with a display behavior. So a lot of people anecdotally hypothesize that this orange coloration is probably an anti-predatory function. Any predator that's about to eat a turtle pretty typically knows to flip it over and go for the softer belly. Their plastrons are ossified later in life, if you're talking about hatchlings, and in general, it's just easier to get to kind of the soft tissue of the limbs if they're upside down. So in order to determine whether this orange coloration has an anti-predatory function, one of the first things I did was construct visual models to determine how highly contrasting painted turtle hatchlings would be, which is when they're most often eaten, to common predators. So I used 3 different birds and a raccoon, all of which are ready predators of hatchling turtles. And not surprisingly, found that to all of these predators, that orange coloration is really obvious against a dirt background or against a grass background. And this is an actual nesting site. So it's probably pretty relevant. However, that doesn't tell us anything about what their actual behavior is or how they neurologically process it. So then I conducted some experiments where I made these models. These are made out of a really soft plasticine where the paint chipped pretty much as soon as you touched it, annoyingly. And I made some orange models, orange with this kind of black plastral shape approximation, tan and tan with a black plastral shape. And I made thousands of these and put them out at 7 different lakes and measured predation rates. So here's the results of that, and this is, notice on the x-axis, starts at 90%. And essentially there was no difference, basically, in how often they were attacked. They were all attacked. I had a lot of data to collect, thankfully, and they were all attacked pretty equally. So what this told me is that it doesn't seem like that orange color has an anti-predatory function at all. So this made me have to really kind of step back from thinking about this turtle system and try to wonder, why in the world would they have this presumably energetically costly orange coloration that they move into their shell, they obtain from their diet, that they don't use as a sexual signal or as an anti-predatory signal. And so this probably would occur to a lot of other people because we've heard it in a few talks today, which is great. But pigments can have other functions besides just making coloration. And to me, when I was an early grad student, this was really surprising to learn. But many pigments, including the flavonoids we heard Matt talk about and Julianne, melanins, and carotenoids, can all have antioxidant functions because they're able to oxidize and reduce other groups. And the reason that antioxidants are important is because reactive oxygen species, also called ROS, are produced as a natural byproduct of most cellular respiration and pretty much a lot of cellular processes. However, if the amount of ROS outweighs the amount of antioxidants, that results in what's called oxidative stress. And oxidative stress can cause lipid peroxidation, cell death, tissue death, and eventually organismal death if it gets too bad. So antioxidants are really important to combat ROS in any given situation, especially during costly events like reproduction, immune challenges, or anything like that. ROS are also more likely to become overwhelming in extreme or dynamic environments. So urban environments, there's been a lot of oxidative stress studies in the last 10 years from comparing urban and rural environments. Here's the Greenland ice sheet, deep sea areas, changing areas, or like a constantly changing tidal zone. All of these could be potentially environments that make the organisms prone to oxidative stress if they don't have equivalent antioxidant protection. And so one interesting thing that's really one of the painted turtle's coolest skills is that they are freeze tolerant. So this species up here, both of these two species are extremely freeze tolerant. Sorry, this is all one species. Both of these subspecies are extremely freeze tolerant. This one is a little bit, this guy is not at all. And so what this means is during the winter, especially their first year of life, in the first year of life, they stay in their nest and they freeze solid. Ice forms, heartbeat stops, blood stops flowing, everything. When spring comes, they recover and they're fine. As adults, the rest of the time, they do a really similar adaptations, but it's actually instead of tolerating freezing, they're tolerating anoxia. So they seek out anoxic hibernacula, where there's no oxygen, their blood stops flowing, their heart beats like once every few minutes. It's really equivalent adaptations to freezing the rest of their adult life. And both of these, here's painted turtle hatchling with ice formation. He's frozen solid. So here's where antioxidants might come into play and might explain that variation in pigment that we're seeing. Antioxidants are going to be especially important anytime blood is cut off from tissue and then reintroduced. So for instance, in human stroke, when a clot forms, let's say that clot breaks down, the person's okay, but the damage from the clot breaking down and blood being reintroduced, which is called reperfusion injury, can often be higher than or cause more damage than the initial event itself. And this is because anytime oxygen is cut off from tissue and reintroduced, everything basically goes into overdrive to make up for what's called the oxygen debt. And this is not super well understood physiologically why this happens, but we know that when everything kicks into overdrive, when blood's reintroduced and oxygen's reintroduced, a lot more ROS are produced than normal. And so in this case, antioxidants are going to be incredibly important in order to mitigate any oxidative stress and prevent reperfusion injury. So this has a lot of relevance to human health and in strokes or heart attacks or transplant surgery, anything that involves cutting off blood from tissue and then reintroducing it. We have to be able to combat the ensuing oxidative stress after that point. So in order to investigate whether these carotenoids might be relevant antioxidants for mitigating oxidative stress after freezing, I took a bunch of hatchlings and put them through a simulated winter. So for the first few months, all of this time they're in complete darkness to simulate the nest environment. They have no access to food or water, but the temperature is decreasing steadily. And then here they were frozen solid for 36 hours. And they also, this is a whole different thing, but they can also supercool. So we tap them with ice chips to make sure they actually froze solid, just because those are two slightly different processes. So this is a turtle in a tube with a thermo data logger. You can barely even see him there because he's very frozen. So we held them there for 36 hours, and during this whole time, we measured oxidative stress and also the carotenoid chroma of the plastron. So I was really lucky to have this, to form this awesome collaboration at Dartmouth with the Electron Paramagnetic Resonance Spectroscopy Center. It's a mouthful, but just call it the EPR Center. And the EPR Center was primarily developed to measure singlet oxygen and free radicals in humans. So it's used for radiation therapy, or this particular lab used it for cancer oximetry, so they would measure the oxygen in a tumor. But they were really happy and really willing to just let me throw some turtles and frogs and other organisms in there to be able to get an in vivo measure of oxidative stress. So that probe that's pointed there, there's a little turtle in there, the probe is pointed at the tissue of interest, and without killing or invasively removing any tissue, you can measure the oxidative stress at that time of that organism. So I was able to measure oxidative stress all the way through freezing, or all the way up until freezing. I didn't touch them for the 36 hours. They were completely frozen because they had ice formation. And then immediately after. I also, with the help of this lovely undergraduate assistant, made sure to establish this relationship, or made sure that there was this relationship between carotenoid chroma, which we can measure non-invasively just by using reflectance spectrometry on the plastron, with the actual carotenoid concentration in the plastron. And you can see it's not a super strong relationship, but it does give us an ability to predict how many carotenoids are in that shell based on the color of it. So here is on the x-axis, we've just broke it down into periods. This is pre-freeze, immediately post-freeze, and then after a week of recovery at room temperature. And on the y-axis, we've got oxidative stress. And this is part of a parallel study where we also froze wood frogs, which are another freeze-tolerant vertebrate. But I included this just to show kind of proof of concept. Like, this is what we'd expect in a freeze-tolerant organism, where they have this pretty high level of oxidative stress early on. And this is, remember, they've gone months without food or water at this point with lowered metabolism. And in both organisms that are freeze-tolerant, we see high levels of oxidative stress. Immediately post-freeze, it's lower, and post-recovery, there's almost none. And then if we look at the carotenoid results here, again, it's broken down into, this is before the cold process began at all, in the midst of it, right before freezing, and then after recovery. We see that these carotenoids start up really high. They have a lot of carotenoids, as you'd expect, based on that orange color. And then it decreases. The mean decreases a bit more, but we have a lot of variance right before freezing. And then after recovery, they're there. We did actually measure them a week later, but the data got ruined, annoyingly. But I know that it was pretty much back to normal. So while none of this is nail-in-the-coffin proof that carotenoids, that the function of this orange coloration is to store carotenoids, this is pretty much circumstantial evidence that they might be mobilized out of the shell to these relevant tissues, like blood and the organs, where oxidative stress is most likely to occur when oxygen is reintroduced. There's also already a mechanism for the movement of things in and out of the turtle shell. We know that during freezing, one of the adaptations they have is they can move out calcium carbonate from the bone and put lactic acid into their shell, and that's one of the ways that they tolerate the effects of anoxia and freezing. And there's also a similar organism in some ways. This is a salmon who stores these carotenoids within the muscle, and then during their intensive reproductive upstream voyage, they pull the carotenoids out of their muscle as they need them to combat oxidative stress. So it's a similar way of storing these carotenoids when you don't need them and pulling them into tissues when you do. And there's a reason for that too, and it's because carotenoids are context dependent, their oxidant activity, where if they're around other antioxidants, they're going to act as antioxidants. But if they're not, if they're alone, they can act as pro-oxidants, which can be damaging. So it makes a lot of sense for the majority of the year for these turtles to keep the carotenoids bound up in this keratin matrix in their shell, where they're unlikely to do any harm, but they're also not likely to benefit. And then during freezing, they're able to pull them out of the shell and use them to combat oxidative stress. So again, this is not conclusive evidence, but this is my working hypothesis, and this is all the work that's been done on this so far. But I want to leave you on one last note, just to keep you interested, I guess, that these guys, so these are all individuals from one population in northern Wisconsin. So this is not the complete picture. Even if carotenoids do have a definitive role in mitigating oxidative stress after freezing, there's still quite a bit of variation, even within a population. And one thing I want to point out really quickly is that the carotenoids are present underneath these melanin patterns. So for what that's worth. But I'm currently in the process. This summer will be my 9th year of collecting long-term field data. It's one of the downfalls of working with turtles is they're so long-lived. It's really hard to get any selection analyses. But I'm doing mark recapture data on this population, so I can determine whether maybe brighter turtles are more likely to survive harsh winters, as well as looking at how that color might change throughout an individual's lifetime, because right now we have no data on that at all. So with that, I want to thank my collaborator with the butterfly work, Michelle Long at Texas A&M, the Kupasami Lab, which is at the Epr Center, and my PhD advisor, Ryan Kelsbeek, and all of these various people and centers for their funding support. Any questions?
So I was wondering if you have any comments on maternal effects. So you figure if they can't produce their own carotenoids and they haven't really eaten anything, then all the carotenoids are packed into the egg. Mm-hmm. Have you thought about having some females in captivity and try to feed them a bunch of carotenoids, or painting the eggs with some or something so that you could do a more manipulative experiment about
I'm so glad you asked. So 4 of the 5 years of my PhD, I collected eggs from females, which is not easy because you have to find them when they're walking the roads about to nest. And I injected the eggs with carotenoids, different levels, trying to raise them and see if they had lower oxidative stress and would tolerate freezing better. Every one of those years, all those experiments failed. So I have no data on that. In regards to maternal effects, one of the things that got me interested in this at all was wondering why hatchlings of freshwater turtles are often so much more brightly colored than adults. And in painted turtles, they're one of the few species that doesn't get less brightly colored as they get older. But if you even look at a hatchling turtle, if you found it just come out of a nest, it's bright yellow right around where the yolk sac is. So they are directly absorbing those carotenoids from their mother, and that's definitely where they start. We don't know if they're actively maintained, like if they have to be eating this diet to get carotenoids. But anecdotally, turtles in captivity that are fed commercial diets or bad commercial diets will often lose color because they're not getting as many carotenoids. So it does appear that it has to be actively maintained. Yeah.
You mentioned that carotenoids are context dependent, that they change their chemical conformation to fulfill different requirements for the turtle as needed. Is it only those 2 requirements that they fulfill, or are there other possible conformations that haven't been found yet, or possible uses that you only suspect but haven't been able to find proof for yet?
Sure. So as far as I know, they don't actually change their chemical composition. It's just that they'll act differently. They'll gain or lose an electron if there are other antioxidants present, basically because they need those other antioxidants to jump off of. So they'll quench that other antioxidant, and that antioxidant can then have the effect. So they're almost more of antioxidant boosters, I guess might be a better way to put it. But they're all also, carotenoids in a lot of organisms have shown other functions besides just antioxidants. We've shown a non-specific ability to increase immune function that we don't know how they're doing it, basically, or why the carotenoids matter. And then in a lot of systems, especially birds, we've looked at how relevant antioxidants are or carotenoids are as antioxidants and have found they're almost not at all relevant in a lot of systems. So it's really interesting that we know they have this ability. We don't know exactly what context it works really well as an antioxidant in.
Yeah. Well, we want it on recording. Impressive running, Tanya. Really nice talk, just knocking down hypotheses one by one. I want to be annoying and give you one more.
Please.
This reminds me of the copepod literature, which is completely new to me. I just discovered it lately, and they had this big debate about why copepods produce carotenoids. And one of the things that was, all of the ones that you've talked about were thought about, but in addition was that the role of carotenoids as energy storage molecules, which, as you can imagine, in a difficult winter, might be beneficial. So the carotenoids are long chains of carbon, as you say, very expensive for that reason, but also might produce, be a nice sort of food storage.
Yeah.
So, sorry, but there you go.
No, that's an interesting point. But I would think, well, firstly, I guess I don't understand the full physiological mechanism of what that energy storage is, so I'm definitely going to look into it. But I think that we would then see that decrease in carotenoids during the cold acclimation, which we did see some. But I would expect it to be a lot more and not to continue decreasing right after freezing.
I think
so
the
U shape, yeah.
When I was saying that, I was thinking the Ross seemed more likely, but they're not mutually exclusive.
No, yeah, definitely, you're right. That's why I'd be interested in the physiological mechanism of that, because it might actually have something to do with this exchanging of electrons that makes them have energy. So thank you for that.
The turtles are fascinating, but I want to ask about the butterflies.
Sure.
Something I hadn't thought about, but of course the wing must be the same shape, so that the two functions, they're working within the same space, and then some of the pattern shows through, that if there's a strong ring on one side, there's something on the other. So within what color changes can happen, were there certain things besides the total amount of change that were highly variable? Were there edges or middles, or where would the really places that could be painted differently?
Yeah.
Is any feel for that?
So along with collecting all these reflectance spectrometry measures, I also took photographs so that I can later go back and do pattern work. From what I could see, the patterns weren't super visible, and I only worked with specimens that were in pretty good condition, so the scales weren't rubbed off at all. If scales were rubbed off, you could definitely see on the other side, and I'm not sure how, not to say that you can't, but I'm not sure how relevant that is in a natural setting, so I'd really have to look into pattern a bit more. That's a great point, because the dark bands are very obvious, and they are working within the same shape constraint, so that's interesting, definitely. Yeah.
Hi, I'm also wondering about the butterfly story, which is that you sort of presented some evolutionary models for the evolution of colorfulness. I'm wondering if those traits are actually sort of more specified, different patches of color within the wing surface, how that would affect your interpretations of sort of the patterns you saw.
Yeah, so I'm really excited to continue to do this kind of species-specific work, and by species-specific, I guess I mean clade-specific. I can break it down into a little bit more generalizable. Like I showed, those 20 that I showed out of the 50 were not cherry-picked. Like they just really are that diverse. So it'll be interesting to look at clades where I can hopefully narrow down some pattern or even just wing shape and be able to look in more detail at the different measures of color, like even orange brightness or any of that. I have all the data to be able to pull out specific things that aren't just colorfulness. This is just where I started from the big macroevolutionary approach. But yeah, that's a great point.
So going back to the turtles,
yes.
You showed that this is not a mating signal, the red.
Most likely.
But I'm wondering if somehow it's related to mating success through condition. because these redder individuals would
Yeah.
be in better conditions because they resist the freeze better.
Yeah.
So I'm wondering if you have a measure of mating success across these populations and if you can relate it to color.
Yeah, I wish, but I don't. So my long-term mark recapture data is pretty much just mark recapture. I've been measuring color for also, I think, for 8 of the 9 years of these data. And I also have, one nice thing about turtles is that you can look at their plastron and they have these age rings on the scute, which they fade after time, but the difference between the rings can tell you a bit about how much they grew that year. So I can get growth out of it. I can't necessarily get fecundity data. Eventually, like if I get a big enough team, it would be amazing to be able to find nesting females and mark their nests and track that. But at this point, it's going to be mostly just looking at survivorship and growth through time, and determining if that's related to color at all. Yeah. Awesome. Thank you so much.
Thank you.